Hello everyone, I'm Hui Zhang from Auburn University. Today I will talk about how to integrate GWAS and principal component analysis to study the mean flavor-related traits in cultivated peanut. First, I will give some background information about peanut flavor. Peanut flavor is a complex trait affected by raw material and processing technology, and it is an important research area due to its significant influence on consumer preference. Peanuts can be consumed in different formats, such as peanut butter, peanut oil, roasted peanut, and nutritional bars. While the primary and popular consumption is prone to the roasted peanuts because of its unique, pleasant flavor and abundant nutrients, although it can amenable to various processes. Genome-wide association study, short for GWAS, is an examination of many common genetic variants in different individuals to see if any variant is associated with a trait. For example, two groups, one is the health group, the other one is disease group. They will be sequenced together and then the variance will be calculated using statistics method. If one type of the variant is more frequent in peanut with a disease, the variant is said to be associated with the disease. The associated variants are then considered to mark a region of the peanut genome that may influence the disease. GWAS typically focus on associations between single nucleotide polymorphisms and traits like major disease, but it can equally be applied to any other genetic variants and any other organisms. Principal component analysis is an effective method to extract major information from complex components. It uses dimensionality reduction method to transform multiple correlated phenotypes to a smaller set of uncorrelated variables as principal components. Therefore, the aims of this research include to explore correlations among sensory and quality attributes to identify the main traits that have major effect on peanut flavor, to determine genomic regions that are associated with main peanut flavor traits, and to identify potential candidate genes residing within the identified QTLs. For the technical line, the original 33 traits were did a Pearson correlation analysis. One trait was excluded due to it has no significant relationship with the other traits. PCA was conducted for the left 32 traits, and after condensed PCA, 3 mean flavor-related traits were left and were used for GWAS analysis. For GWAS analysis, we need both phenotype data and genotype data. The concentrations of 3 traits of 102 US peanut mini-core collection were obtained through 2 years experimental repeats. The genotype data was got from SNP array. 2 methods were used for GWAS analysis. The first one is TASO with the PCA and the GLM model, and GAPIT with the ML model as the second method. According to the QQ plot, finally TASO was chosen for GWAS analysis. This slide shows the 102 US peanut mini-core collection, the population structure, and the LD decay. From this picture, we can know the mini-core collection will mainly include 2 groups. and the LD decay in this population was approximately 160 kilobase, and the R square was 0.2. The broad sense heritability of most traits are very high, such as oil-contained, total tocopherols, and total sugars. The heritability is stable, that's why we only use the 2 years data. According to the condensed PCA, the first 6 principal components for 13 traits were picked considering the normal distribution of the phenotype data of the traits. Finally, 3 traits were chosen for GWAS analysis, including total tocopherols, sucrose, and total sugars. Other 3 traits, roast peanut, metallic, and astringent, also have the big principal component data, but the phenotype of these 3 traits is not belong to the normal distribution, so we choose the first 3 used for the GWAS. In general, the concentrations of these 3 traits in different environments was consistent, and the phenotypic data displayed a near normal distribution. A total of 67, 91, and 20 non-redundant QTLs were detected for total sugars, sucrose, and total tocopherols separately, among which there were 7 significant QTLs for total sugars and 22 for sucrose. However, significant QTLs were not identified for total tocopherols. This slide shows Manhattan and QQ plots. The red horizontal line indicates the genome-wide significant threshold is 4.67. The blue line indicates the threshold for suggestive association is 3.37. From these Manhattan plots, we can note that the significant QTLs are mainly on chromosome 14 and chromosome 15 for total sugars and sucrose. For tocopherols, there is no significant QTLs above the red line. 2 stable significant QTLs associated with total sugars were identified in both years, and both of them explained 16.13% of the phenotypic variance. Similarly, 3 stable significant QTLs located at chromosome 14 and 2 stable significant QTLs located at chromosome 15 were detected related to sucrose. 2 significant QTL regions on chromosome 15 were found associated with both total sugars and sucrose. The genes nearby significant and suggestive QTLs within 160 kilobase regions were detected. A total of 4 potential candidate genes associated with total sugars nearby significant QTLs were detected, including 1 stable gene, BXRG0F, which is located in a genomic region on chromosome 15, encoding nucleotide diphosphor sugar transferase superfamily protein. The functional annotation of the other 3 genes in significant QTL regions include that the 2 genes encode UDP glycosyltransferase, and one gene encodes SC-CoA. For sucrose, a set of 8 genes were identified within the significant associated region. The stable gene BXRG0F related to sucrose was also detected in both 2 years. In addition, 4 genes nearby significant QTL regions are associated with both total sugars and sucrose, which includes the one stable gene BXRG0F. Discussion: peanut flavor is a complex process which is affected by genetic, environment, and storage conditions. Both sample size and SNP numbers can affect the ability to achieve significance. Further RT-PCR or gene knockout experiments will be needed to verify the candidate QTLs or genes. In conclusion, the relationships among 33 traits affecting flavor were studied by Pearson correlation. GWAS was conducted for the main 3 traits which have major effects on peanut flavor: 2 stable QTLs and 1 stable gene BXRG0F were identified controlling both total sugars and sucrose. Thank you for your attention.